Don't do title slides. I'm just going to bail right in. Um, uh, First, I'd like to thank the organizers for inviting me. I had a really interesting conversation at lunch. And I do come from the old-time molecular biology genomics background and into the world of cancer, having spent a sabbatical year at the National Cancer Institute last year helping to set up the Center for Cancer Genomics. And so that's the perspective and how I come to know a bit about this topic. In cancer, the big data questions, the science of it, and also the service of providing big data to researchers and to the wider community has become really central just because of the nature of the problem scientifically. Oh, I'm so non-PC in every sense.
So you can either double click it to advance it, or you can just use that.
Thank you. I'll stick here. Thank you. So there's always the question, and I'm sure that you have it in the environmental community also, when you're thinking about generating big data, are you generating it to answer mainly one hypothesis and then you want to make it available to others? Are you generating big data on faith because you think that there will be lots of ways to mine it? Is it something in the middle? And who's really your target audience? So with respect to cancer at NCI and NIH, there's the internal intramural community. There is a host of others in concentric circles out around the big data and the tools for big data that go on out to the Center for Cancer Genomics, and then it goes to the over 50 cancer centers in the country, then to extramural cancer biologists, and ultimately to an even broader community than that. So here's a way to think about cancer genomics and the decade roughly 2008 to 2018, which, talking about history and talking about what we project forward, is the rough range that I'm going to be covering. So there's discovery by genomics and functional genomics, and that's a fundamental, of course, because cancer is essentially a genetic disease based on somatic and germline mutation. All of these things, we hope when we get the data and use it right, will influence drug development, identification of pathway functions, DNA-based diagnosis, and all of that will land in a precision treatment and also in prevention. Now, something that's become a new and major cause that interests me and other people in the room is how can we get back huge amounts of information from the patient population, even above and beyond the trial system, coming out of diagnostics done with DNA and coming out of treatment and outcomes back into the research and discovery phase. And as you'll see in a moment, and as most of you are aware, the underlying science drives the need for dealing with numbers of individuals, and that means numbers of genomes and exomes and RNA-seqomes and transcriptomes and so on and so forth, that are even much larger than our current biggest big data projects. Okay, so there's the history of what's been done that I'll talk about at the outset, some obstacles that have come and lessons that we think we've learned. There was a large project that covered, depending on how you count its endpoints, about a decade at NCI that ultimately got mixed reviews, some very successful aspects and some really painful lessons learned. The TCGA, the Tumor Genome Atlas project, which is coming toward its completion and is, I think, widely regarded as successful and increasingly so. And I'll say something just very briefly, since you and Bernie will talk soon about ENCODE. I've got just one little example from the ENCODE consortium. And then the future needs and expectations. Okay, first I'll start with TCGA and give you some recent history coming out of it. What does it do so that you can gauge what we know from it? It does multiple data types from multiple sources. It's taken on 25 forms of cancer. It gathers many data types. Of these data types, a subset that are highlighted here in red are sequence-based, and they therefore come in for the special treatment that involves controlled data access, which is a non-trivial part of our enterprise, and though it's not a focus for this group, I am going to urge that it be in your minds at all times when you're making big decisions, because it surely will affect the utility of your data and who can use it and how they can use it. Provenance of samples. There are 6 genome characterization centers, 3 sequencing centers, 7 analysis centers, a data coordinating center. Provenance and handling of the samples and of the data, primary and secondary processed, is a huge undertaking in something of this size, which is 500 samples for each kind of cancer. So the driving problem, we have to have the tools and we have to have proper data access in order to compute over all the data types and all the sources. And so it's that integration enterprise, and I'm just as sure as I'm standing here, I know that Ewan will have things to say about integration, about multiple data types. But that's certainly what drives many of the additional requirements on a database and on the tools that need to be developed. This is what the pipeline looks like for TCGA, beginning with tissue sample at the upper left and proceeding all the way down to integrative computational analysis and ultimately a comprehensive characterization of these cancers that takes into account their endpoints as well as their genomics. Now, there are 2 points. The very beginning point, the collection of the samples, which is rate limiting and incredibly costly. The other point, this end process of handling the data, storing the data, doing the integrative analysis, and ultimately extracting real knowledge by lots of researchers, including people who didn't participate in gathering the data, that turns out to be the other very costly, rate limiting, and ultimately value limiting piece. So what do we do with TCGA data when we get it? Currently, data storage is in a special database. This has been set up under UC Santa Cruz, led by David Hausler, under contract to NCI. This is a big enterprise to hold the TCGA on target genomics projects. For those who are into the details, it stores all the primary files as well as much derived data. It has both open access for derived data that's no longer judged to be identifiable, and it has controlled access currently under the dbGaP model. And we can talk more about that if somebody is interested at the end. This is a download mode only database. In other words, you don't compute over the data at the data center. This is a database much like NCBI is a database. It's designed for about 25,000 cases with an average of 200 gigs per case. Some anticipated compression is optimistically in there. And we have a mutation in the advance on this, so it's not terribly important. So the point is, we have a big system, and that big system, I'm going to tell you in a few minutes, has reached its limits of scalability. And we need to move to something different that isn't only bigger, but is materially different in its type. And a good deal of thought has gone into that, and that's an exciting enterprise for the immediate and downstream future. But once you've got data storage, we've got something that will handle TCGA and target right now. The next question is, what constitutes successful use? And I think one of the biggest problems with projects that has been handled pretty well in TCGA and target is thinking ahead of time, what use will be success and how will you measure it? Now, one kind of thing you can look at is how many investigators from where are doing how many downloads of your data, and here's a typical such measure for primary sequence files. An interesting fact is that the controlled data that's primary sequence is really important. There's big time science going on with that, but that's less than 1% of usage. It turns out that there's an awful lot of use of the process data, and not surprisingly, data spikes after publications. You can follow this closely and see exactly what's going on with your data user community. So there's a different way that you can measure success. This takes a bit more time, and it might be even more important. And that is measuring the kinds of places where people who have gone and reused the data, not to make the primary paper, but have gone to the data, used the data in some creative way often to generate the starting hypothesis for a more directed study that follows. And so this is moving beyond just characterizing the shape of the data and doing primary discovery of, for example, driver genes in the case of cancer, and moving beyond that to the next steps. And this happened to be a nice early study of that sort. Okay, making computational analysis reproducible. This has been a big problem, a big challenge in TCGA target, and we went through some pain to make it more right. It turns out that even when serious professionals engaged in a fairly coherent consortium go to prepare a very large data set from, say, 500 tumors, and they go to prepare it for publication, at the end of the day, it's still the same. There's a key postdoc working in the trenches somewhere. And that key postdoc working in the trenches somewhere is keeping perhaps better or worse track of the number of trials for processing the data. And you want to have one data freeze, but as I know Ewan can say, and Will from ENCODE, well, sometimes there are loose data freezes, then there are secondary data freezes, and so on and so forth. And what you end up with at the end of the day when you think you have been very assiduous is a clear data freeze, data processing, conclusions derived, and you have the software you used, or at least you think you do. But it turns out that unless you make an incredibly concerted effort to capture exactly all the code, all the scripts, and get them out there along with all the primary data and do the experiment yourself that says, if I take all these data at the freeze and I run this software, I get out these figures in the paper. Okay, that's a hard thing to do in a big study, and I don't think that going in people typically appreciate how really hard that is. And for many published papers coming from many laboratories, and I would count some of my own, regrettably, that match is not a perfect match. We thought it was, but it's not, and it's important. So getting dry pipelines and turning them over to people so that they at least have the same starting point that was produced should be a goal, and often it's not a goal except ex post facto when somebody calls up and says, I did it, and it doesn't work. Okay, process evolution in a large consortium can be a really wonderful thing if done right, and it can be a horror if done wrong. Here's an example that I would call doing it right. Again, it comes out of TCGA, and it's the calling of mutations in tumor DNA. So this is one of the main enterprises for sequencing exomes and ultimately whole genomes. It's to be able to call the mutations, determine the frequency of the mutations, decide which things occur above statistical significance and therefore are likely to be biologically causal drivers of that particular kind of cancer or subset of cancers. Okay, now at the beginning, the computational pipelines for the participating centers for calling mutations had been derived in exactly the way they should be, by people who generate software and do the coding who are also really users searching for the mutations. And one of my themes in watching these projects, several of the big ones, is that the most valuable people you can have, the most valuable people we can produce in the training enterprise are people who in fact are at this interface and can actually generate the tools because they want to know the answer and they understand deeply the nature, the shape, the quirks, and the warts on the data. That's our dream. There aren't enough of them. We need more, yada, yada. However, what they did in TCGA was say, okay, we're not agreeing. In fact, when they put these mutation calls together, you can look at that Venn diagram, and anybody would go, oh my God. Okay, each one of these centers thought they had a good set of calls of mutations. And while I would bet my life on conclusions derived at the deep center of that Venn diagram, this is a pretty worrisome set of outer sets. So what happened was that a series of benchmark experiments were done, and it took a while to call the first benchmark study and get it done, which was not we're all out processing our own data and we'll get it together at some future date, but right now we're going to take a data set that's blind to everybody, and we're all going to call it. And then we're going to compare, and then we're going to try to figure out partly what's more right, but really just why are they different? Where's the emphasis different? So there have now been 4 of these studies, and they get more and more sophisticated. And, you know, there are obvious things. Doing the calling eventually from a consensus derived from the benchmark studies ends up getting you a better set of mutation calls. The collaboration involved, and this is the argument for multiple participants in a consortium, not a single sequencing center, not a single contractor, is that you really see what the differences are. And this comes up in all of these consortia, probably comes up in yours as well if you're doing a big tox screen. If 2 people do essentially the same set of chemicals but using a completely different high throughput assay and different kinds of cells, my bet is that in the field, which I don't know well enough to answer, that there's an interesting overlap and that there's a really interesting set of outer sets from which one can learn a lot. So this is just a slide to point out that they're now up to benchmark 4. And at this point, the key point in a slide that was made for other purposes is that the wider cancer community, even beyond the initial participants, are now invited to jump in because there are more groups outside of the initial primary ones that are themselves generating mutation callers that are looking at very specific things like structural variation rather than point mutations and so forth. And that's a really good thing. One of the take-home lessons from CA-BIG that they did really, really well was to get lots of participants at the table repeatedly to think about and talk about how to build standards. Not to do the experiments together necessarily, but to talk about what the joint needs are, and there will be abrasion and there'll be argument in that. And if you're not getting abrasion and argument and people in the room whose blood pressure is up, then you're not doing it right. And if you are doing it right, you'll get all of that, but out of that cauldron of seething self-interest will actually come something that can be a really useful core synthesis to work around. And I have the feeling a number of us are kind of trying to learn the history of the early internet, and I think all of us are frustrated that we haven't learned enough yet about how the decisions were made that moved toward common protocols that had commonality at just the right core level without dictating that everybody has to do exactly the same thing all the time and without stifling new innovation that moved to the next thing. And someday, if I ever understand how we got to HTML and XML and so forth, we may have insights that we can apply to our own road. I would just encourage people here to go down that road, and if you get answers, tell me. But I think it's really what we should be trying to do. Out of ENCODE, this is my only ENCODE slide, but I have worked, my laboratory has worked in the ENCODE consortium as part of its endeavors for the past years and made us very aware of what you can get when a consortium goes to work on some kind of standard experiment, and this happens to be the one for locating how proteins bind and are located over the chromosome in living cells by chromatin immunoprecipitation sequencing assays. And we've been very interested in the development of that from the beginning of the wet work through the dry work and so forth. And what I just want to point out is that what the consortium was good for was battling through and looking at a bunch of different programs that different people had invented and kind of doing a bake-off and saying, well, you know, here there are a bunch of good ones, but here are the top 2 or 3, and here are their characteristics, and this one's more sensitive to this kind of background, this one's more sensitive to that. Developing an understanding, then making standards for the consortium that affected the data and the data processing, and saying what they are, not to dictate it to the whole community, but to say, here's a shot on goal, and here are some of the reasons why we do it, how we do it, and here's what we got out the other end, and here's a big data collection that's all been subjected uniformly to this kind of analysis. And then what follows out of that are other endeavors where people go out and look at the public data and they do it. And there's a paper that's been put together that's about that. And the other thing is that it helps for the wider community to get out what they need to know. That is to say, if you looked at a very useful tool, which is an IDR, it's a measurement of how you deal with replicates in this world, which was a statistical problem for us. And, a great statistician came to the rescue, Peter Bickel and his group, and they got the method, and they wrote a paper that statisticians could respect. I challenge any end-user biologist in the universe to go to this paper and figure out what to do with it in their experiments. And so what the consortium mode does is to get somebody like young Anshul Kunde, who actually not only applied this, but very nicely explains it to the world at this website so that people can actually understand, who are not statisticians, what it is that they're doing and why. Okay, future for cancer. Individual tumors, they have lots and lots of different driver mutations. These happen to be lung tumors. This is the biology that drives the need to move from 500 tumors to 500,000 tumors. Metadata. You all are concerned about environmental things. You can see from the little cartoon down on the side, we're looking at rates of mutations and kinds and patterns of mutation and the way in which they vary across cancers. This is organized roughly with the upward trend in density of mutations found sequencing exomes. And you can see the impact where there's a big known environmental carcinogen over on the right-hand side in the lung cancers for smoking, sunshine for melanomas, and so forth. This means lots of metadata. So not only do we see the need for a huge increase, 100-fold or more increase in DNA sequence data, but an equal increase in the complexity and richness of metadata. And it is that which is going to drive changes in the nature of both the tools to work with the data and integrate over all these variables, and also the need for a different kind of database. EMRs, everybody wants the information from them. It isn't from scanned PDFs. Are we ever going to be including primary images? Just think what that's going to do to database requirements. And I leave you to consider that. So what comes in the future? What's going to come in the future in this leap by 10x and 100x comes the prediction that we're going to be talking about 1 million exomes and whole genomes by just 2015. Oh, maybe it'll be 2016. The British have committed themselves to 100,000 over the next 2 or 3 years. There are clearly commitments and large-scale studies ramping up in the U.S. And there is the important idea of drawing information as sequencing becomes a commonplace in the clinic back where the research enterprise can actually get it. That should happen, and it should be able to happen in this country. I would personally argue it's not clear that it will. We have the scientific problems to solve. We have the regulatory problems to solve about access and use. And we have the problems of our not integrated medical care system. This is going to be done in the cloud, certainly. It has to be done in the cloud. It's not only going to be storage in the cloud, it's also going to be computing over the data in the cloud. And the reason is that even if we had the money, even if we made a gigantic room with more and more and more things like our current database, the problem is the upload-download time. And the problem is the amount of compute that everybody has to have at home if they're going to download it, and if they're going to compute over all of it, they've got to have huge compute capacity. We, the aggregate community, clearly cannot afford that. Interoperability is something that CA Big went after, and it's huge, and we have to have it, and it does call for periodically getting the stakeholders in the room and talking about how interoperability will be achieved, and it needs to evolve. It can't be static. I guess we all know these things intuitively, but what we see is that when we go to do small projects, medium projects, and gigantic projects, the fact that we know this does not always translate into doing even approximately the right thing. Crowdsourcing is very attractive, by the way, and clearly that will happen if you go to one place to compute and if you have reasonable, not too restrictive standards so that people can develop knowing what they're developing for. Okay, the Envision Cancer Genomics Commons continually define the information and computational analysis needs. The idea that you can sit down and do it once and then have it done and just go do it, like you do, I have a hypothesis, now I'm going to go do the experiment and then the answer will come out, is not how it can be. It has to be an ongoing process, but with enough solidity to it that you move from one stage to the next stage rather than doing continuous oozing. And so the problem is to get the mix right so that you're evolving, but not in a continuous ooze so that you never have anything stable. And that's just the big trick here. There's a tension between research and service informatics. We clearly need both. These are different people and different issues. There's a tension, or at least a distinction that I think is important, between computational analysis and bioinformatics. In cancer and anything else, we know you need both, but too often somebody in some administrative place somewhere or some funding place thinks that if it involves a computer, it's really all the same stuff, and try to get economies by hiring somebody who claims they can do all of these things. And that's a lesson that I think has been learned probably in other venues, but certainly don't go there. You need to recognize who it is you need to solve what problem and what kind of problem you're trying to solve. There's the naive user versus the sophisticated user. Managing expectations of your several communities is clearly something that we've run across continuously. And the idea that a user at the Broad is the same as a user who's the one graduate student who's computer literate sitting in a laboratory in, wherever, they're not. And that the expectations, and particularly the expectations of really interested, the old-timers in any field. They're really interested, they're running successful labs, they're moving to high throughput, they get somebody who's a good informatician and figure the problem is solved and that they will be able to do interfacing with anything out there that involves a computer. You really have to be careful about managing the expectations, not as a way necessarily of currying favor with your community, although that's not too bad a thing to have. It is because everybody simply has to understand what to expect. You're bound to be disappointed if you think that you're going to get, for all that we curse the products out of Redmond, Washington, the fact of the matter is that there are 38,000 people that put together essentially one suite of software. That's not what we have to produce GUI-ized things to solve every problem. You're not going to be able to do that. And having people who use data broadly in a big data sense understand that, who need to interact with big data but are not big data mavens themselves. Best developers are users. Find them, recruit them, make them, fund them. I can't say that enough. And here's a lesson that was definitely learned in the sort of first very big scale thing that NCI did in this venue, which is avoid letting technical developer groups, doesn't matter if they're contractors or colleagues or who they are, but if a group of them all get together and just talk to each other a lot and get the bit in their mouths and run with the agenda and say, what you guys, you don't know what you need. Just go away and we're going to get you what you need. Doesn't work. It's never going to work. Don't go there. I mentioned controlled access earlier. I'll say it one more time. There is a question about who's going to get access to all of these data, as I know that part of the enterprise in this community is certainly the genotype-phenotype exposure relation. And because most sequence-rich data is ultimately, in some sense, re-identifiable, all of the issues that revolve around who should look at that data and what rules and what hoops do they need to jump through in order to look at those data, and will it be seamless? Will it be easy? I think that although this is very central, of course, to the cancer enterprise dealing directly with patients and tumors, that it will branch out as the common rule is reconsidered to include lots of things that affect people in this room. And we must, as a larger community, if I can do anything, it's to simply get this in front of you to know to think about, and to think a little bit deeply about, should someone ask you in the near future how it should be done. It has to be electronically facile. The kinds of things that we have done for very good reason to try and give responsible, protected access to data that relates to informed consent in a very intimate and proper way, where consents have not been harmonized for broad use, leads to data that have a great deal to offer that can't. And they simply cannot be integrated because there are too many little bits and pieces that have to be navigated to draw in each data set. And so we need to find a better way to do this. And there are lots of people who are thinking about this very actively, and I think there are ways that can safely respect both the wishes and the privacy needs of patients, and nevertheless provide real facility for the kind of big data mining that we need to do. And most of the big data mining that we need to do is going to be, I would argue, to good purpose compared to lots of big data mining that's being done on everybody all the time, that is to, shall we say, less certain purpose. And finally, I want to bring to your attention a great resource, which probably many of you know, but some may not. It's the Academy's National Research Council report from November 2011 of the study that was co-chaired by Charles Sawyers and Susan Desmond-Hellman, that's called generically the Precision Medicine Study. But it is a really exciting view forward of what we can do in this country, building a knowledge commons that includes genetic information, clinical information, environmental information, and so forth. And it points out what many of the pitfalls and challenges are bound to be. In the process, I'd like to predict that NIH, the whole of it, may very well turn out to be a modest part of the future medical information commons, because there are other large driver forces at work here with an interest in seeing this happen and happen well. Yeah, I think the suggestion is that for NCI, as big as it is, or for any other institute or program within an institute, that planning forward, being informed of and integrated into this larger context is going to be important. And if one goes forward with something now in complete isolation, that will be too bad. And finally, leadership in big data for biomedicine and environmental studies and so forth. I think a lot of us might mistakenly think that it's our birthright if we just don't screw it up. But actually, in this case, it's very possible that the U.S. will be the trailing edge in a while, not the leading edge. We're in a position to make it the leading edge, but it's by no means assured. Okay, and with that, just kind of a reality check note, I'll take any questions. Oh, Titus.
Well, I was going to say...
This is probably going to be a zinger.
Well, it's not often I get to question you. Barbara was on my...
We can still revoke that PhD.
No,
there's fine print, Titus. There's very fine print.
So I just wanted to mention, and I've already fought this battle a little bit with Ewan on the ENCODE stuff. So there's this interesting distinction in reproducibility between replication and reproducibility. And what you talked about is we can take you use the term a dry pipeline, which I've never heard before. And the data, and you can reproduce the contents of the paper. And I spoke about that in my talk, and I think it's a great starting point. The problem is it doesn't necessarily enable remixing. The idea that I can take the pipeline that you used for your analysis and deploy it on my data.
Oh, it absolutely does not say that. Right.
And I just wanted to say I think that it's a good bar, but it's a low bar for where we should be thinking about
My
computational
experience is that the low bar is missed so often.
Yes.
But
Yeah.
but I do think that the high bar, part of it is whoever's doing the establishment of pipelines number one needs to actually understand it well enough to be able to be even remotely prescriptive or helpful about whether somebody else's data is even a fit to go through that dry pipeline at a low technical level or at a high scientific level, both. As usual, we do agree at the end. Okay.
Okay, so I think we'll